Cancer was found in the bones of a dinosaur 76 million years old. It was found in the fossilized remains of marine organisms from the Cambrian period, 500 million years ago. It has been found in trees, in fungi, in corals, in the shells of ancient mollusks. Cancer did not invent civilization. Cigarettes did not invent it. Poor nutrition did not invent it. Modern life did not invent it. Multicellularity invented cancer. At the very moment when the first cells began cooperating and building bodies, they created the condition under which any cell in any body could betray the contract and begin acting in its own interests. That happened 600 million years ago. And since then, not one species of multicellular organism has found a way to eliminate this completely. Not one, including the ones that tried very hard. Before we go any further, if this kind of deep, unhurried scientific storytelling is worth your time, please subscribe to this channel. It genuinely helps us keep making it. Thank you. Now, to understand what cancer actually is, not in the clinical sense, not in the oncological sense, but in the deepest evolutionary sense, we need to start not in a hospital or a laboratory, but in the pre-Cambrian ocean, approximately 600 million years ago, at the moment when one of the most consequential transitions in the entire history of life on Earth was occurring. A transition so fundamental that it set the biological conditions for everything that followed, for every animal, every plant, every fungus, every complex organism that has ever lived on this planet, including the conditions for cancer itself. For the first 3.5 billion years of life on Earth, every living organism was a single cell. Bacteria, archaea, early eukaryotes, all single-celled, all autonomous, all operating entirely in their own interests. A single-celled organism has no conflicts of interest between its parts. It has no parts. It is a single unit of Darwinian selection. It either survives and reproduces, or it does not. And every molecular process in the cell is aligned with that single goal. There is no cancer in a world of single-celled organisms because there is no cooperation to betray. Then, approximately 600 million years ago, something changed. Cells began to cooperate. Not loose associations, genuine biological integration, in which cells with identical genomes began to specialize, to divide labor, to perform different functions in service of a shared organism whose fitness was distinct from the fitness of any individual cell. Some cells became structural, others became reproductive, others became sensory or digestive or locomotive. The multicellular organism was born, and with it, immediately and inevitably, the conditions for cancer. Here is why. In a multicellular organism, the cells that make up the body share an identical genome. They are all descendants of the single fertilized egg that initiated the organism's development. But they are not identical in their behavior. Through the process of differentiation, the activation and suppression of different genes in different cells, they have become specialized. Liver cells behave as liver cells. Neurons behave as neurons. Muscle cells behave as muscle cells. This specialization is maintained not by genetic differences between the cells, but by epigenetic programs. patterns of gene expression that are stably maintained through cell division and that define the cell's identity and constrain its behavior. The constraints are severe. A differentiated cell in a multicellular organism has surrendered most of its evolutionary autonomy. It does not reproduce at its own discretion. It divides only when the organism signals that division is appropriate, responding to growth factors, wound healing signals, and developmental programs. It does not consume resources without limit. It takes what it requires and no more. It does not migrate freely. It remains in its tissue of origin unless specifically instructed to move. It accepts death, programmed cell death, apoptosis, when it receives signals indicating that it is damaged, aged, or no longer needed. It cooperates, in short, with the other cells of the organism, suppressing its own individual fitness in service of the collective fitness of the body. This suppression of individual cellular fitness is the evolutionary foundation of multicellularity and the precise location of its vulnerability. It carries a genome that can mutate. If a mutation gives a cell a growth advantage, if it allows the cell to divide more rapidly, to ignore the signals that normally restrict division, to resist the apoptotic signals that would normally eliminate it, that cell will, through the logic of natural selection, expand. It will produce more descendants than its neighbors, and those descendants will carry the same mutation and potentially accumulate additional mutations that further enhance their growth advantage and expand further still. This is cancer, not a disease in the conventional sense of an external pathogen invading the body, not a malfunction in the sense of a machine breaking down. Cancer is evolution. Darwinian evolution by natural selection, operating within the body, producing cell populations that are progressively better adapted to the local tissue environment and progressively worse aligned with the cooperative requirements of the multicellular organism. The tumor is, in the most precise biological sense, an evolving ecosystem within the body. Its cells vary genetically. They compete with each other and with normal cells for resources. They are subject to selection, those variants best adapted to survive the local environment, including the environment of chemotherapy or immune attack, proliferate while less adapted variants are eliminated. The tumor evolves, and because it evolves, it adapts. And because it adapts, it is extraordinarily difficult to eliminate permanently. The evolutionary perspective on cancer is not merely an interesting intellectual framework for understanding a disease. It is the framework that explains why cancer has proven so resistant to the most sophisticated therapeutic arsenal that human science has ever assembled. We have spent the last 50 years approaching cancer primarily as a problem of molecular biology, identifying the specific mutations that drive specific cancers and developing drugs that target the molecular products of those mutations. This approach has produced genuinely remarkable achievements. The treatment of chronic myeloid leukemia with imatinib, the treatment of BRAF mutant melanoma with targeted inhibitors, the treatment of HER2 positive breast cancer with trastuzumab. These are real advances that have extended and saved many lives. But the limits of this approach are visible in the pattern of treatment failures. Targeted therapies that are initially highly effective at shrinking tumors often lose efficacy over time, sometimes within months, as the tumor evolves resistance. The drug eliminates the sensitive cells. The resistant cells, which were present as a minority population before treatment began, are now freed from competition and expand. The tumor regrows, not as the original tumor, but as a new evolutionary variant, selected specifically for its ability to survive the drug that was supposed to eliminate it. The treatment created the very enemy it was designed to defeat through the mechanism of natural selection. This is not a failure of pharmaceutical chemistry. It is a failure to account for the evolutionary nature of the disease. You cannot win a Darwinian arms race by applying selective pressure. You will always select for the survivor, and the survivor will be better adapted than anything you faced before. Understanding this is the beginning of a genuinely different approach to cancer treatment, one that takes the evolutionary nature of the disease seriously and designs treatment strategies that work with evolution rather than triggering evolutionary escape. But before we can understand what a truly evolutionary approach to cancer treatment might look like, we need to understand the deeper evolutionary story. The story of how multicellular organisms across the animal kingdom have addressed the cancer problem over hundreds of millions of years of selection, and what the extraordinary variation in cancer rates between species tells us about the biological mechanisms that suppress cancer most effectively. The fundamental puzzle at the heart of cancer biology, the paradox that gives this field some of its most intellectually productive tension, was articulated in 1977 by the British epidemiologist Richard Peto, and it is known, simply and directly, as Peto's paradox. The paradox can be stated simply: large, long-lived animals should have much more cancer than small, short-lived ones, but they do not. The logic behind the prediction is impeccable. Cancer arises from mutations in somatic cells, cells of the body, as opposed to the germline cells that transmit genetic information to offspring. These mutations arise during DNA replication, when errors in the copying process introduce changes in the DNA sequence. They arise from environmental damage, radiation, chemical mutagens, reactive oxygen species from metabolism. And they arise from the normal aging process as the cumulative burden of DNA damage accumulates in cells over time. Every cell division carries a small but non-negligible probability of producing a cancer-initiating mutation. More cells means more cell divisions means more opportunities for cancer-initiating mutations. Longer lifespan means more time for mutations to accumulate in any given cell. A blue whale has approximately 1,000 times more cells than a human being. A bowhead whale, the longest-lived mammal, surviving for over 200 years, has both more cells and far more time in which those cells can accumulate mutations. The mathematics of somatic mutation theory predicts that blue whales and bowhead whales should have cancer rates that are orders of magnitude higher than those of humans or mice. They do not. Cancer mortality in elephants is approximately 4%, compared to approximately 25% in humans. Blue whales and bowhead whales appear to have cancer rates that, while not zero, are far below what their size and lifespan would predict. Naked mole rats, which live for 30 years, over 7 times the lifespan expected for a rodent of their body size, show essentially no spontaneous cancer in study populations that have been followed for decades. Peto's paradox is not a paradox in the logical sense. It does not represent a genuine impossibility. It is a paradox in the empirical sense, a prediction that follows clearly from established theory is not observed in nature, and the resolution of the paradox reveals something profound about the evolutionary biology of cancer suppression. The resolution is this: large, long-lived animals have not escaped the elevated cancer risk that their size and lifespan would create. They have evolved countermeasures, enhanced cancer suppression mechanisms that keep that risk in check despite the larger number of cells and longer lifetime over which mutations accumulate. And the diversity of those countermeasures across different species is a treasure trove of biological solutions to the cancer problem that evolution has been developing for hundreds of millions of years. The elephant story is the most extensively studied and the most dramatic. Elephants have 20 copies of the tumor suppressor gene TP53, a gene that, in humans, exists in a single copy and is mutated in over 50% of all human cancers. TP53 encodes the p53 protein, often called the guardian of the genome, a transcription factor that is activated by DNA damage, and that, depending on the severity of the damage and the cellular context, either initiates cell cycle arrest, pausing division until the damage can be repaired, or triggers apoptosis, programmed cell death, eliminating the damaged cell entirely before it can accumulate the additional mutations that would lead to cancer. In humans, with a single functional copy of TP53 per cell, the cancer suppression system can be disabled by a single mutational event that inactivates the single functional copy. In elephants, with 20 copies of TP53, 19 of which are retrogene copies, derived from a reverse transcription event that inserted a processed copy of TP53 mRNA back into the elephant genome, probably multiple times during elephant evolution. Disabling the cancer suppression system requires many more mutational events. An elephant cell that suffers DNA damage is far more likely to undergo apoptosis than a human cell with equivalent damage. The elephant TP53 system is, in effect, a much more sensitive and much more redundant damage detection and elimination system. This is a 2025 paper from the study by Leonard Nunney, published in the journal Evolutionary Applications, which re-examined the role of elephant TP53 retrogenes with greater rigor, found that the evidence for the functional significance of the TP53 retrogenes in cancer suppression is robust, and that their evolutionary acquisition tracks the evolution of large body size in the elephant lineage. The retrogene copies appear in the elephant lineage at approximately the time when the ancestors of modern elephants were evolving toward larger body sizes, consistent with the hypothesis that the cancer suppression system was enhanced in response to the increased cancer risk that larger body size created. The elephant also has 11 extra copies of a gene called leukemia inhibitory factor, or LIF. One of these copies, LIF6, is a reactivated pseudogene that has evolved a new function. It is activated by p53 in response to DNA damage, and when activated, it enters mitochondria and triggers the leakage of the mitochondrial membrane, initiating cell death. This is a second independent apoptotic pathway activated in response to DNA damage, a redundant cancer suppression mechanism that works in parallel with the direct p53 apoptosis pathway. The elephant has, in effect, evolved belt and suspenders cancer suppression. If p53-mediated apoptosis fails, LIF6-mediated apoptosis provides a backup. The practical implications of this are already being explored. Joshua Schiffman at the University of Utah, one of the primary researchers of elephant TP53, has founded a company called Peel Therapeutics, from the Hebrew word for elephant, that is exploring the delivery of elephant TP53 retrogene sequences to human cancer cells via nanoparticles, with the goal of making human cancer cells as sensitive to DNA damage as elephant cells. The approach is in very early stages. This is described as early days research even by Schiffman himself. But the conceptual logic is sound, and the elephant has provided the evolutionary proof of concept. The naked mole rat provides a completely different evolutionary solution to the same cancer suppression problem. And the contrast with the elephant solution is illuminating about the range of biological strategies that natural selection has explored. Heterocephalus glaber, the naked mole rat, is a small, hairless, eusocial rodent native to the dry savannas of East Africa that lives in underground colonies of up to 300 individuals with a reproductive queen, non-reproductive workers, and a hierarchical social structure that has no parallel in any other mammal, except the closely related Damaraland mole rat. It lives for over 30 years, more than 7 times the expected lifespan of a rodent of its body size. And in study populations that have been followed for decades, covering thousands of individuals, spontaneous cancer has been essentially absent. The naked mole rat's cancer resistance is not mediated by enhanced TP53 or by enhanced apoptosis sensitivity in the way that the elephant's is. It operates through a fundamentally different mechanism, the extracellular matrix. The tissues of the naked mole rat are extraordinarily rich in a molecule called high molecular weight hyaluronan, HMW-HA, a large polymer of the extracellular matrix that plays multiple structural and signaling roles in tissue organization. The HMW-HA produced by naked mole rat cells is dramatically larger in molecular weight than the hyaluronan produced by the cells of other mammals, including mice and humans. And this unusually large hyaluronan activates contact inhibition, the cellular response to crowding that signals cells to stop dividing when they are surrounded by too many neighboring cells, at much lower cell densities than in other species. Contact inhibition is one of the most fundamental cancer suppression mechanisms in multicellular organisms. It is the mechanism by which normal cells in a healthy tissue sense that the tissue is sufficiently populated and reduce their proliferation accordingly. When a wound heals, cells at the wound edge proliferate to fill the gap, then stop when they contact neighboring cells. Contact inhibition terminating the healing response. Cancer cells, by contrast, have lost contact inhibition. They continue to proliferate even when surrounded by neighbors, ignoring the crowding signals that would normally arrest their division. The naked mole rat's HMW-HA system activates contact inhibition at a much earlier stage of crowding, effectively preventing the initial cell population expansion that would be required for a tumor to form. The gene responsible for producing HMW-HA in the naked mole rat, HAS2, carries specific mutations that distinguish it from HAS2 in all other mammals, producing a version of the enzyme that generates longer hyaluronan chains. When researchers replaced the normal mouse HAS2 gene with the naked mole rat version in transgenic mice, the mice showed reduced cancer rates and extended healthy lifespan, demonstrating that the naked mole rat HMW-HA system can function as a cancer suppressor in a different mammalian species. The bowhead whale, Balena mysticetus, provides a third evolutionary solution, operating at the level of DNA repair, rather than apoptosis sensitivity or contact inhibition. The bowhead whale is the longest-lived mammal known, with maximum lifespans estimated at over 200 years, based on the analysis of harpoon tips found embedded in living whales, tips manufactured in the 19th century, and confirmed by molecular clock analysis of eye-lens proteins. A 200-year-old bowhead whale has had 200 years of cell divisions and metabolic DNA damage accumulating in its cells, an amount of mutational burden that should, by the calculations of somatic mutation theory, guarantee cancer. And yet, bowhead whales appear to have cancer rates far below those predicted by their size and lifespan. Genomic analysis of the bowhead whale genome reveals a pattern of duplications and variations in genes involved in DNA repair and cell cycle regulation. The molecular machinery that detects and corrects DNA damage before it can accumulate into the mutational burden that initiates cancer. The bowhead whale appears to have enhanced the fidelity of its DNA repair systems over evolutionary time, reducing the rate at which somatic mutations accumulate in each cell division, and thereby reducing the probability that any given cell will accumulate the multiple mutations required for full malignant transformation. This is not a perfect solution. No cancer suppression system is perfect, but it is a highly effective one that has maintained a large-bodied, exceptionally long-lived whale in reasonably good oncological health for over 200 years. What the elephant, the naked mole rat, and the bowhead whale collectively demonstrate is that evolution has multiple independent solutions to the cancer suppression problem, that the biology of cancer resistance is not constrained to a single mechanism, but can be achieved through apoptosis enhancement, through contact inhibition enhancement, through DNA repair enhancement, and presumably through many other mechanisms that have not yet been characterized in detail across the full diversity of long-lived, large-bodied animals. This diversity of solutions is not merely academically interesting. It is a catalogue of biological strategies that human biomedical research has only begun to systematically investigate as potential therapeutic targets. Now let us look more carefully at the evolutionary dynamics of cancer itself, at what happens inside a tumor as it develops, grows, and eventually metastasizes. Because understanding these dynamics is essential to understanding why current treatments so often fail, and what a more evolutionarily informed approach might look like. The January 2026 study from the Moffitt Cancer Center, published in Nature Communications, developed a new computational method called Alpha-K that reveals the fitness landscape governing how cancer cells gain and lose whole chromosomes, a process called chromosomal instability that is one of the primary mechanisms by which tumors generate genetic diversity. The study found that chromosome gains and losses in cancer cells are not random. They follow consistent patterns across different cancer types that reflect the underlying fitness effects of gaining or losing specific chromosomes in specific cellular environments. Some chromosomal changes are consistently beneficial for tumor cells across diverse cancer types. Others are consistently detrimental. And the patterns of selection on chromosomal composition reveal the hidden rules of tumor evolution, the evolutionary pressures that are shaping the genetic landscape of the tumor as it grows. This study represents a significant advance in our ability to predict the evolutionary trajectory of tumors, to anticipate which genetic changes are likely to occur as the tumor evolves, rather than merely cataloging the changes that have already occurred by the time a patient is diagnosed. And the ability to predict tumor evolution is the prerequisite for designing treatment strategies that stay ahead of it. The concept of adaptive therapy, developed primarily by Robert Gartenby and colleagues at the Moffitt Cancer Center over the past decade, is the most developed example of a treatment strategy that explicitly accounts for the evolutionary nature of cancer. The central insight of adaptive therapy is that the goal of cancer treatment should not be to kill as many cancer cells as possible, as quickly as possible. The approach of maximum tolerated dose chemotherapy, but to maintain stable control of the tumor at a size that does not threaten the patient's health, by exploiting the evolutionary dynamics of the tumor rather than fighting against them. The evolutionary logic of treatment failure is the same logic we have already described, but adaptive therapy proposes a different response to it. Adaptive therapy, by contrast, applies treatment at doses calibrated to maintain a stable population of drug-sensitive cells in competition with drug-resistant cells, rather than eliminating the sensitive cells entirely. The sensitive cells, kept alive at reduced numbers by lower dose treatment, compete with the resistant cells for resources, limiting the expansion of the resistant population. The tumor is held in a stable evolutionary equilibrium, rather than being pushed toward resistance by the selective pressure of maximum dose treatment. Early clinical trials of adaptive therapy in metastatic prostate cancer, published by Gartenby's group in Nature Communications in 2020, showed that patients on adaptive therapy protocols maintained control of their cancer for significantly longer than patients on standard continuous treatment, with lower cumulative drug doses and fewer treatment-related side effects. This approach is counterintuitive. It deliberately leaves cancer cells alive. It accepts the continued presence of the tumor in order to avoid the evolutionary escape that aggressive treatment precipitates. It manages cancer as a chronic condition rather than attempting to cure it outright. And it is, from an evolutionary perspective, precisely the right approach. It works with the evolutionary dynamics of the tumor rather than triggering evolutionary countermeasures. The immunotherapy revolution has added another dimension to the evolutionary picture of cancer treatment. The immune system is, in evolutionary terms, the primary cancer suppression mechanism in vertebrates, the system that detects and eliminates abnormal cells before they can develop into tumors. The checkpoint inhibitor drugs, the anti-PD-1 and anti-CTLA-4 antibodies that have transformed the treatment of melanoma, lung cancer, and multiple other malignancies, work by releasing the brakes on the immune system, allowing T cells to recognize and attack cancer cells that have evaded immune surveillance by activating inhibitory pathways. The results in some patients have been extraordinary. Durable, long-term remissions in cancers that were previously uniformly fatal, achieved without the severe toxicities of chemotherapy. But immunotherapy also faces the evolutionary challenge. Tumors evolve resistance to immune attack through multiple mechanisms: downregulation of MHC molecules that present tumor antigens to T cells, upregulation of alternative immune checkpoint pathways that immunotherapy drugs do not target, recruitment of immunosuppressive cells to the tumor microenvironment. The immune system exerts an evolutionary pressure on the tumor, and the tumor responds, as all biological systems respond to selection pressure, by evolving to evade it. The co-evolutionary arms race between the immune system and the tumor is not a new phenomenon. It is the same arms race that the immune system has been running against pathogens for hundreds of millions of years. And just as pathogens have evolved extraordinarily sophisticated mechanisms for evading immune detection, tumors evolve similar mechanisms, using the same repertoire of molecular strategies. The liquid biopsy revolution, the detection of tumor DNA, fragments circulating in the blood of cancer patients, offers a window into tumor evolution in real time that was not previously available. Cell-free tumor DNA carries the mutations present in the tumor at the time of sampling, and serial liquid biopsy analysis can track the appearance and expansion of new mutations, including resistance mutations, as the tumor evolves during treatment. This allows treatment decisions to be made on the basis of the current evolutionary state of the tumor rather than the state at the time of initial biopsy, weeks or months earlier. The tumor has changed. The treatment can change with it. Now we come to the question that the evolutionary perspective on cancer ultimately forces us to confront. Is cancer curable in the absolute sense? Is there a world in which cancer is eliminated as a clinical problem? Or is it an irreducible consequence of the biology of multicellular life that medicine can manage but never eliminate? The honest answer, from an evolutionary perspective, is that complete elimination of cancer as a disease is probably not achievable through any conceivable biomedical intervention. For the same reason that death from aging is not eliminated by any intervention that merely slows the aging process. Cancer is not an external threat to multicellular life. It is an internal property of multicellular life, arising from the same cellular machinery that makes multicellularity possible. As long as cells divide, they will accumulate mutations. As long as they accumulate mutations, some of those mutations will confer growth advantages. As long as some mutations confer growth advantages, natural selection will favor the expansion of the cells carrying them. This is not a failure of biology. It is biology. What is achievable and what the convergence of evolutionary insight, computational biology, immunology, and molecular medicine is making increasingly tractable is the transformation of cancer from a frequently lethal disease into a chronic, manageable condition. Not cured in the sense of eliminated, but controlled in the sense that the evolutionary dynamics of the tumor are understood well enough, and the therapeutic tools sophisticated enough that patients can live with cancer rather than dying from it, in the way that patients with HIV, diabetes, or hypertension now live with conditions that a generation ago were uniformly fatal or severely life-shortening. This reframing from cancer as a disease to be cured to cancer as an evolutionary process to be managed requires a fundamental shift in how both medicine and patients think about the disease. The expectation of cure, the complete elimination of all cancer cells, has been the primary goal of oncology for decades, and it has driven genuinely important advances. But it has also led to treatment strategies, maximum dose chemotherapy, complete tumor eradication, that are evolutionarily counterproductive, selecting for resistant populations rather than maintaining stable control. The expectation management that a more evolutionarily informed oncology requires is not a counsel of despair. It is a recognition that managing cancer intelligently, using the evolutionary dynamics of the disease rather than fighting against them, may produce better outcomes than the scorched earth approaches of maximum dose therapy. The Santa Fe Institute workshop of January 2026, organized around the theme of cancer as a failure of multicellularity, brought together evolutionary biologists, cancer biologists, and mathematical modelers to develop a more rigorous theoretical framework for understanding the evolutionary transitions that produce cancer and the evolutionary transitions that suppress it. The key insight emerging from this work is that multicellularity requires not just the evolution of cooperative cellular behaviors, but the evolution of mechanisms to enforce those cooperative behaviors against the constant evolutionary pressure toward defection. Cancer is what happens when the enforcement mechanisms fail, and the diversity of enforcement mechanisms evolved across the animal kingdom, from elephant TP53 to naked mole rat hyaluronan to bowhead whale DNA repair, represents a billion-year archive of evolutionary solutions to the same enforcement problem. The September 2025 paper in Evolution Medicine and Public Health, proposing a sixth universal foundation of multicellularity, proximity maintenance, builds on this framework. The 5 previously identified foundations of multicellularity are proliferation inhibition, controlled cell death, resource allocation, division of labor, and maintenance of the extracellular environment. The 6th, proximity maintenance, The tendency of cells to remain in close contact with their neighbors rather than migrating freely maps directly onto one of the most clinically consequential hallmarks of cancer: invasion and metastasis. Metastasis is not merely the spread of cancer cells to distant sites in the body. It is the breakdown of proximity maintenance, the evolutionary dissolution of the spatial organization that defines the tissue and that cells of the tissue are committed to maintaining. Understanding metastasis as the failure of an evolutionarily ancient cellular behavior, rather than as a late-stage complication of a localized disease, has implications for how we think about prevention and early detection. The genetic changes that enable metastasis may be targeted earlier in the disease process, before the full metastatic program has been activated. The molecular signals that maintain proximity, the adhesion molecules, the extracellular matrix interactions, the contact inhibition signals, may be therapeutic targets that can be reinforced before they fail, rather than replaced after they have already broken down. The cancer suppression mechanisms evolved by elephants, naked mole rats, and bowhead whales are not curiosities of exotic biology. They are demonstrations provided by evolution across hundreds of millions of years of trial and error that the cancer suppression problem is solvable. Not perfectly solvable, not permanently solvable, but substantially improvable beyond the level that the human lineage has achieved. We have one copy of TP53 where the elephant has 20. We have standard-sized hyaluronan where the naked mole rat has supersized hyaluronan. We have a DNA repair system of a certain fidelity where the bowhead whale has a more accurate one. Each of these differences is, in principle, addressable by biomedical technology that is currently in development, or at least in conceptual reach. The fossil record of cancer is one of the most unsettling and most illuminating bodies of evidence in the entire evolutionary story of the disease. When we say that cancer is ancient, that it predates human civilization, predates mammals, predates vertebrates, we are not speaking metaphorically or extrapolating from molecular biology. We are speaking from direct physical evidence. Tumors preserved in stone, in amber, in fossilized bone, across hundreds of millions of years of geological time. The oldest unambiguous evidence of cancer in the fossil record comes from the Cambrian period, approximately 500 million years ago. Fossilized shells of marine invertebrates from this period show abnormal tissue growths that paleontologists have interpreted as neoplastic lesions. Uncontrolled cellular proliferation producing structural distortions in the shell architecture that are inconsistent with normal developmental programs. These are not injuries, not parasitic infestations, not taphonomic artifacts of the fossilization process. They are the preserved signatures of cells that broke the cooperative contract of multicellularity 500 million years before any vertebrate animal existed. The dinosaur evidence is particularly striking because it connects the deep evolutionary history of cancer to organisms whose biology was, in many respects, closer to our own. A study published in 2020 by Natalia Jagielska and colleagues documented an osteosarcoma, a malignant bone tumor of the type that most commonly strikes human adolescents during rapid skeletal growth. In the fossilized femur of a Centrosaurus apertus, a horned ceratopsian dinosaur that lived approximately 76 million years ago in what is now Alberta, Canada. The tumor was initially misidentified as a healed fracture, and its true nature was only recognized through detailed three-dimensional micro-CT scanning and comparative histological analysis against a reference collection of modern human osteosarcoma specimens. The pattern of abnormal bone remodeling, the disorganized trabecular architecture, the evidence of cortical invasion, all were consistent with osteosarcoma in a modern human patient. The dinosaur osteosarcoma is not the oldest cancer in the vertebrate fossil record. Benign bone tumors have been identified in the skeletons of hadrosaurs, the duck-billed dinosaurs, at frequencies suggesting they were genuinely common pathologies in living dinosaur populations. Evidence of cancer-like lesions has been found in the fossil vertebrae of Permian-era synapsids, the mammal-like reptiles that preceded the dinosaurs and from whose lineage mammals eventually evolved, pushing the vertebrate cancer record back to approximately 250 million years ago. Plants provide a completely different window into the universality of cancer across multicellular life. Crown gall disease, caused by the bacterium Agrobacterium tumefaciens, which injects a segment of its own DNA directly into plant cells, hijacking the plant's cellular machinery to produce uncontrolled cell proliferation, produces tumors on plant stems and roots that are, in all biologically relevant respects, cancers. The infected plant cells proliferate without the normal regulatory constraints of plant tissue organization, producing a disorganized mass of cells that consumes the plant's resources and disrupts its normal architecture. Crown gall has been studied extensively, not merely as a plant pathology, but as a model system for plant genetic transformation. The same Agrobacterium gene transfer mechanism that causes crown gall has been adapted as the primary tool for introducing foreign genes into plants in agricultural biotechnology. What crown gall demonstrates is that the cancer problem, the vulnerability of multicellular organisms to uncontrolled cell proliferation, is not specific to animals. Plants evolved multicellularity independently from animals, using different molecular mechanisms and different regulatory architectures. Yet they face the same fundamental challenge: maintaining the cooperative discipline of billions of cells in the face of the constant evolutionary pressure toward individual cellular defection. And they are, like animals, vulnerable to failures of that discipline when the regulatory architecture is disrupted, whether by bacterial DNA insertion, by viral infection, by radiation, or by the same accumulation of somatic mutations that drives animal cancer. The universality of cancer across the full diversity of multicellular life, in Cambrian marine invertebrates, in Cretaceous dinosaurs, in Permian synapsids, in 21st century Tasmanian devils, and Tasmanian devil's ancestral lineage, in oak trees infected by crown gall bacteria, in soft-shell clams dying from transmissible leukemia, is the most powerful statement of what cancer actually is. It is not a disease of civilization. It is not a consequence of pollution or processed food or sedentary lifestyle, though all of these things can increase cancer risk. It is a fundamental property of multicellular life, as old as multicellularity itself, as universal as the cooperative contract that multicellularity requires, and as inevitable as the evolutionary pressure that continuously tests the boundaries of that contract. The dinosaur in the Alberta Badlands 76 million years ago did not know it had osteosarcoma. The Cambrian marine invertebrate, with its neoplastic shell lesions 500 million years ago, had no awareness of the cellular rebellion occurring in its tissues. But the rebellion was real. The evolutionary logic that produced it was the same logic that produces cancer in a human patient today. And the story that connects that Cambrian organism to that dinosaur to that patient, the story of multicellularity's ancient vulnerability and evolution's imperfect but persistent response, is the story we have been telling here. The atavistic theory of cancer is one of the most conceptually striking frameworks to emerge from the intersection of evolutionary biology and oncology in the past 2 decades. And it reframes the question of what cancer actually is in a way that has profound implications for both treatment strategy and our understanding of the deep evolutionary history of the disease. The theory was developed primarily by the physicist and cosmologist Paul Davies and the oncologist Charles Lineweaver, and it proposes something that initially sounds almost paradoxical. Cancer is not a random breakdown of cellular machinery producing chaotic, disordered growth. Cancer is a reversion, a return to an ancient, highly organized program of cellular behavior that predates multicellularity, preserved in the genomes of multicellular organisms for hundreds of millions of years, and reactivated when the suppressive mechanisms of multicellularity fail. Consider what cancer cells actually do. They proliferate without limit, ignoring the signals that restrict division in normal tissues. They resist apoptosis, evading the programmed death signals that eliminate damaged cells. They induce the growth of new blood vessels to supply themselves, angiogenesis. They invade surrounding tissues and migrate to distant sites, metastasis. They reprogram their metabolism to rely on glycolysis, even in the presence of oxygen, the Warburg effect. They evade immune surveillance. They create local environments that suppress the immune response and support their own growth. Each of these behaviors, considered individually, looks like a malfunction, a breakdown of the normal cellular order, but considered collectively, they form a coherent, highly organized program that is recognizable from a different biological context. The behavior of single-celled organisms operating in a competitive environment. An amoeba proliferates without limit when conditions allow it. An amoeba resists death. It does not undergo programmed death in response to the signals that kill differentiated cells of a multicellular organism. An amoeba migrates toward nutrient sources. An amoeba does not cooperate with neighbors in the way that cells of a multicellular tissue do. The behavioral repertoire of a cancer cell is, in a very precise sense, the behavioral repertoire of a successful single-celled organism. The atavistic theory proposes that this is not coincidental. The genes that control the hallmark behaviors of cancer, the oncogenes and tumor suppressors that are the primary targets of cancer mutations, are among the most evolutionarily ancient genes in the human genome. When researchers analyzed the evolutionary age of genes associated with cancer across the entire human genome, they found a consistent pattern. Cancer driver genes, the genes whose mutation most consistently promotes cancer, tend to be older genes, present in unicellular ancestors before the evolution of multicellularity. The newer genes, those that evolved specifically with the evolution of multicellularity, tend to be tumor suppressor genes, the genes that enforce the cooperative constraints of the multicellular body. Cancer, in this framework, is not the activation of new, dangerous genetic programs. It is the deactivation of the regulatory layer that multicellularity imposed on top of ancient unicellular programs, revealing the unicellular default state that was always present beneath the multicellular surface. The tumor is not chaos. It is order, but the order of an ancient single-celled organism, no longer constrained by the cooperative requirements of the multicellular body. This reframing has a therapeutic implication that is beginning to be explored in experimental oncology. If cancer represents the reactivation of a specific, organized ancient program, then the genetic state of cancer cells, the combination of oncogene activations and tumor suppressor inactivations that characterizes a given tumor, may not be as random as the catalogue of cancer mutations suggests. There may be a limited number of stable ancient cellular programs that cancer cells can revert to, and these programs may have characteristic vulnerabilities that could be exploited therapeutically. A treatment strategy that targets the ancient unicellular program itself, rather than the specific mutations that deactivated the multicellular regulatory layer, might be more broadly applicable and less vulnerable to evolutionary escape. Because the ancient program is itself conserved and less able to mutate away from its characteristic vulnerabilities without losing its own functional integrity. The Warburg effect, the preference of cancer cells for glycolytic metabolism even in the presence of adequate oxygen, which the German biochemist Otto Warburg identified in the 1920s, and which is one of the most universal metabolic features of cancer, fits naturally into the atavistic framework. Glycolysis is a far more ancient metabolic pathway than oxidative phosphorylation. It predates the endosymbiotic acquisition of mitochondria by eukaryotic cells, and it is the primary energy-generating pathway of many single-celled organisms. A cell reverting to an ancient unicellular program would be expected to upregulate glycolysis at the expense of oxidative phosphorylation, not because glycolysis is more efficient in terms of ATP production per molecule of glucose, but because it was the metabolic default of single-celled ancestors long before mitochondria existed. The cancer cell's metabolism is ancient metabolism, running on ancient pathways, in a body that evolved to run on something far more sophisticated. The relationship between cancer and aging is one of the most consequential and most paradoxical in all of evolutionary biology, and it illuminates the deepest constraints on what any cancer suppression system can achieve. The paradox is this: the strongest cancer suppression mechanisms we know of Mechanisms like the p53 pathway also drive aging. The genes that protect us from cancer also, through their normal operation, accelerate the decline of our tissues over time. This is not an accident. It is an evolutionarily expected consequence of the same logic that we discussed in the context of Peto's paradox. And understanding it is essential to understanding why simply borrowing the elephant's 20 copies of TP53 and installing them in a human being would not be the straightforward improvement it might initially appear. The concept of antagonistic pleiotropy, proposed by George Williams in 1957 as an explanation for why aging exists, holds that many genes are maintained in the population because they confer benefits in early life, even though they are harmful in later life, because natural selection acts much more strongly on early life fitness than on late life fitness. A gene that dramatically improves reproductive success in early adulthood but causes cancer or tissue degeneration at 60 will be strongly favored by selection, because the early benefit accrues during the period of maximum reproductive output, and the late harm accrues after most reproduction has already occurred. The TP53 gene is a textbook example of antagonistic pleiotropy applied to cancer suppression. P53 suppresses cancer by triggering apoptosis or cellular senescence, permanent cell cycle arrest, in cells that show signs of DNA damage or abnormal growth signals. This is enormously beneficial in terms of cancer prevention. The damaged cell is removed before it can accumulate the additional mutations that would lead to malignancy. But the same apoptosis and senescence that prevent cancer also deplete the stem cell pools of tissues that require continuous renewal. Every time a stem cell undergoes P53-mediated apoptosis or senescence in response to DNA damage, that is one fewer stem cell available to regenerate the tissue. Over decades, this depletion accumulates. Tissues become less able to regenerate after injury. Organ function declines. The very mechanism that protected against cancer throughout early and middle life has, by late life, depleted the regenerative capacity of the tissues it was protecting. This is the cancer aging trade-off. More aggressive cancer suppression comes at the cost of faster tissue aging, and slower tissue aging requires accepting greater cancer risk. The trade-off is not theoretical. It is observable in the comparison between P53 hypomorphic mice, which have reduced D53 activity and show reduced signs of aging but elevated cancer rates, and P53 hyperactive mice, which show reduced cancer rates but dramatically accelerated aging. Nature has not found a way to have both full cancer suppression and full tissue maintenance simultaneously, because the mechanisms that achieve one compromise the other. The elephant's 20 copies of TP53 provide enhanced cancer suppression at the cost of more aggressive apoptosis in damaged cells, a trade-off that is acceptable for an animal that has evolved a metabolic and cellular physiology suited to it. But that might not be simply transplantable to a human metabolic context, without producing unintended consequences for tissue maintenance and aging. This is why the development of TP p53-based cancer therapies, like those being explored by Peel Therapeutics, requires careful characterization of the full biological effects of enhanced p53 activity in human tissues, not merely its effects on cancer cells in isolation. The senescence component of p53 activity adds another layer to the cancer aging paradox. Cellular senescence, the permanent cell cycle arrest that p53 can trigger as an alternative to apoptosis in damaged cells, is an important short-term cancer suppression mechanism. A cell that cannot divide cannot develop into a tumor. But senescent cells do not simply stop and do nothing. They secrete a cocktail of inflammatory molecules, the senescence-associated secretory phenotype, SASP, that promotes tissue remodeling, wound healing, and immune surveillance in young organisms. In aged organisms, the accumulation of senescent cells that have been created by decades of p53-mediated damage responses produces chronic low-grade inflammation, inflammaging, that drives essentially every major age-related disease, including, paradoxically, late-stage cancer promotion. The p53 system creates senescent cells to prevent cancer, and the accumulation of those senescent cells eventually creates a tissue environment that promotes cancer. The cancer aging paradox is not a design flaw in the biological system. It is an inevitable consequence of the evolutionary trade-offs that govern multicellular life. Natural selection cannot optimize simultaneously for cancer suppression, tissue maintenance, and longevity without limit, because the mechanisms that achieve each of these goals partially compromise the others. The best it can do, and what it has done, across hundreds of millions of years and thousands of species, is find local optima that balance these competing requirements in the context of the specific life history, body size, and ecological circumstances of each lineage. The elephant's optimum looks different from the human's optimum, which looks different from the naked mole rat's optimum, which looks different from the bowhead whale's. Each is a solution to the same fundamental problem, calibrated to a different set of evolutionary constraints. The most unsettling dimension of the evolutionary story of cancer is the existence of transmissible cancers, tumors that have escaped the bodies of their hosts and become independent, self-reproducing lineages of cancer cells that transmit from individual to individual like infectious diseases. These are not cancers caused by infectious agents, not HPV-associated cervical cancer or Helicobacter pylori-associated gastric cancer, in which an infectious organism triggers cancer in the host. These are tumors whose cells are themselves the infectious agent, living cancer cell lineages that have evolved the ability to survive outside their original host, colonize new hosts, and evade the immune responses that would normally reject foreign cells as non-self. The Tasmanian devil facial tumor disease is the most extensively studied example, and one of the most dramatic wildlife disease crises of the 21st century. Sarcophilus harrisii, the Tasmanian devil, is a carnivorous marsupial native to the island of Tasmania that has been devastated since the mid-1990s by a transmissible cancer that spreads through biting, a behavior common in devil social interactions. The tumor grows in and around the mouth and face of affected animals, ultimately interfering with feeding and causing death by starvation within months of appearance. The disease has killed over 80% of the Tasmanian devil population since its discovery, driving the species to the brink of extinction, and it remains one of the most serious conservation emergencies in Australian wildlife management. What makes devil facial tumor disease biologically extraordinary is that the cancer cells transmitting between individual devils are a single clonal lineage derived from the Schwann cells of a single devil that lived in the 1980s, that has survived as an independent cellular lineage for decades, reproducing by transmission from host to host. The cells are genetically distinct from all living Tasmanian devils, carrying a unique karyotype, a characteristic chromosome arrangement that identifies them as the original clonal tumor. When these cells are transmitted to a new host through a bite wound, they evade rejection by the host's immune system through multiple mechanisms, including the downregulation of MHC molecules that would normally mark them as foreign and trigger an immune response. More recently, a second transmissible devil facial tumor, a genetically distinct clonal lineage, has been identified in Tasmanian devil populations, indicating that the conditions that allowed the first transmissible cancer to arise and spread are not unique and may recur. Two independent transmissible cancer lineages in the same species within decades of each other suggests that the evolutionary transition from a normal tumor to a transmissible cancer may be less improbable than previously assumed. The canine transmissible venereal tumor, CTVT, is even more extraordinary. It is the oldest known lineage of cancer cells in the world, a single clonal lineage that has been transmitting between dogs, wolves, and other canids through sexual contact for approximately 11,000 years. The original animal from which CTVT descended lived approximately 11,000 years ago, somewhere in Central Asia or East Asia, based on genomic analysis, and was a dog, or possibly a wolf, with a genome that shows characteristics of early domesticated dogs. Since then, the tumor cells have been transmitted from dog to dog in an unbroken cellular lineage, surviving for over 100 centuries, while the individual dogs in which they temporarily reside live and die. The CTVT genome carries over 2 million somatic mutations accumulated during 11,000 years of transmission, a remarkable record of the ongoing mutational process in a cancer lineage that has been running continuously since the early Holocene. CTVT is, from an evolutionary perspective, one of the most remarkable organisms on Earth. A mammalian somatic cell lineage that has evolved into an effectively independent organism, reproduced by direct cell transmission rather than by DNA transmission through a germline. It has a body, the tumor, a means of reproduction, sexual contact between dogs, and a lineage that has persisted for 11,000 years across millions of individual host animals. Whether it qualifies as a distinct species in any meaningful biological sense is a genuinely interesting philosophical question without a clear answer. The discovery in 2015 that soft-shell clams on the Atlantic coast of North America were dying from a leukemia-like disease caused by a clonally transmitted cancer cell lineage, and that similar transmissible cancers existed in multiple other bivalve mollusk species, extended the phenomenon of transmissible cancer from vertebrates to invertebrates, and suggested that the evolution of clonally transmissible cancer may be a more common evolutionary event than the limited number of characterized examples implies. Marine bivalves, living in dense populations in filtered seawater through which dispersed cells might easily travel, may provide an environment particularly conducive to the evolution of cellular transmissibility. How many other transmissible cancer lineages exist in the natural world, in species whose health has never been systematically monitored, is entirely unknown. The transmissible cancers make viscerally concrete what the evolutionary framework for cancer implies in the abstract: that cancer cells, under the right selective conditions, can evolve beyond their original role as defectors within a single multicellular body and become independent biological entities, pursuing their own evolutionary interests across the bodies of multiple hosts. The cancer cell's ancient single-celled program, unlimited proliferation, resistance to death, migration, immune evasion, turns out, when elaborated by thousands of generations of selection within a tumor environment, to be capable of producing something that functions as an independent organism. The cancer cell wants, in the evolutionary sense, to persist and reproduce. And in a very small number of cases, the Tasmanian devil, the dog, the clam, it has found a way to do so that transcends the lifetime of any individual host. This is the ultimate expression of the evolutionary logic of cancer, the cooperative contract of multicellularity, betrayed not merely within a body, but between bodies. The single-celled program not merely reactivated, but liberated. The tumor not merely growing, but traveling. It is, simultaneously, the most frightening possible expression of the cancer phenomenon, and one of the most scientifically illuminating, because it demonstrates, with stark clarity, what cancer cells are evolutionarily trying to do, and how far the evolutionary process can take them when the selective environment permits. The deeper evolutionary lesson is this: cancer is the price of complexity. Every multicellular organism that has ever existed has paid this price in some form. The organisms that have paid it most successfully, that have evolved the most effective cancer suppression systems, are the ones that faced the highest cancer risk from their size or lifespan, and that therefore experienced the strongest evolutionary pressure to develop countermeasures. The elephant's 20 copies of TP53 were not a gift. They were the product of millions of years of selection against cancer in a large-bodied lineage in which cancer mortality was a significant fitness cost. We are, as a species, at a moment when we have the scientific tools to read this evolutionary archive, to understand what the elephant's genome, the naked mole rat's extracellular matrix, and the bowhead whale's DNA repair system are telling us about the biology of cancer suppression, and to begin translating those evolutionary solutions into biomedical interventions. The translation is not yet complete. Peel therapeutics is in early days. The naked mole rat, HAS2 gene has been tested in mice, but not in humans. The bowhead whale DNA repair mechanisms have been identified but not yet pharmacologically exploited. But the direction is clear, and the evolutionary logic is sound. Cancer was invented by multicellularity 600 million years ago. It has been the subject of evolutionary countermeasures in lineage after lineage, independently across the entire tree of animal life, ever since. The human lineage has developed cancer suppression mechanisms that are, by any reasonable measure, quite good. We suppress cancer through most of our lives, and the median age of cancer diagnosis in the United States is around 66 years. But we are not the best at it. The elephant is better. The naked mole rat is better. The bowhead whale is better. And understanding why they are better at the molecular, cellular, and evolutionary levels is the scientific project that is most likely to produce the next generation of genuinely transformative cancer therapies. The dinosaur bone with its fossilized tumor tells us that cancer was not waiting for us specifically. It was waiting for anyone who built a complex body and filled it with dividing cells. Every complex organism that has ever lived has participated in this ancient biological negotiation between the cooperative requirements of multicellularity and the evolutionary imperatives of individual cells. We are not special in this regard. We are merely the species that has become sophisticated enough to understand the negotiation, to read its terms in the genomes of elephants and naked mole rats and bowhead whales, and to begin, carefully, imperfectly, but with genuine and accelerating insight, to renegotiate. The cancer in the dinosaur bone was not a failure. It was a consequence. The same biological logic that built the dinosaur, the cooperation of billions of cells in service of a complex body, also created the conditions for the tumor that grew in its bone. The same logic built you, and the same evolutionary process that spent millions of years equipping elephants with 20 copies of TP53 has now, in us, produced a species capable of reading those copies, understanding their function, and beginning to use that understanding in the service of the most ancient health problem that complex life has ever faced. The price of complexity is real, but so is the intelligence that complexity eventually produces. The intelligence that is now, for the first time in the history of multicellular life, looking back at the problem and beginning to work out how to pay less of it.